Okay, so I figured it might be easiest for me to do just a video recording of how I typically do the CD68 data quantification on Fiji. That way, you can like go back to this when you're looking at it, and it's a lot of going back and forth between my Excel spreadsheet and Fiji, but very straightforward. And since you already have Fiji experience, you will have no problem with this. So I went ahead and already made two Excel spreadsheets because we're going to want to quantify two things. First, the number of microglia. So I set up an Excel spreadsheet to do that here. And then we're going to want to quantify the number of CD68 positive microglia. So this is just the number of microglia that have CD68 puncta inside either the cell bodies or the microglial processes. And I'll show you on image J how we determine that. But basically, I set up these Excel spreadsheets that make data analysis a lot easier for me. And pretty much any sort of like quantification that I do on Fiji, I will use this spreadsheet, and I will I have it as a template saved on my Excel, and I just duplicate it and change it as needed based on what I'm quantifying. So first here, I've written down the names of each mouse. So I have mouse 1 LPS. This one is male 1 LPS. So with females, I usually do F1, F2, F3. How you label it is up to you. This is just how I usually label. And then we're going to have 3 sections per mouse. So as we did with the IHC, and with the confocal, we took images of 3 different sections, and we're going to average those together. So that's going to be our value for that mouse. I always put the region I'm looking at. So this is the somatosensory cortex. The region of interest area, which is important for our density quantification, and I'll show you in just a second how I get that value. Microglia, I've left blank because we're going to go through that now as we quantify. The Z, this is important. So as you remember, when we took images on the confocal, we took a Z stack, which is the thickness essentially of each slice, and we did a Z value of 25 microns, and we chose a Z step size of 1. So that means that the microscope is going to take an image every 1 micrometer through this entire 25 micron thick stack, if that makes sense. So the Z is actually going to be 26 because it starts at 0. So it's essentially like it's going to take an image at 0, and then it's going to take an image at 1, 2, 3, all the way to 25 until it covers the thickness of the entire 25 micrometer thick stack. And if you remember, we kept that constant for each slice because when we're going to quantify the microglia and the CD68, we're going to make a projection where it basically superimposes all the cells we see in the entire stack onto one screen. And so it's important to keep the Z the same because if you, for instance, if I looked at a 100 micrometer thick slice, that's probably going to have a lot more cells than one that's 25 micrometers thick. So it's important to keep note of the Z. Then in this column, I have the area times the Z stack because, again, we're looking at slices, which are not just in the X and Y plane, but they have a thickness. So we're going to get the volume. And this is the A times Z in micrometers cubed because that's the area that it gives us is in micrometers, but we want to convert this to millimeters cubed to get a density quantification. And so that I just plugged in this equation of multiplying whatever is in this this column by 10 to the negative 9. And then the density that we're going to find. So once I have the quantity of microglia, which we're going to count in a second, I'm going to divide that by this density. So that'll tell me I have this many microglia in this essentially little like cube of density that I'm looking at. And then this right here is just taking the average for each of the 3 sections for each brain region. And so the values that are in this I column are ultimately going to be the ones that we plug in to do statistics and make our graph. So I'll share this with you. Same thing for the CD68 quantification, except instead of just all microglia, what we're going to quantify is the number of microglia that have CD68 puncta inside of them. So now I will go ahead and go to Fiji and kind of walk you through how I do this. So let me open up Fiji. Okay, so this is open. So I'm going to go to this July 26 folder. And let's go ahead and start with the M1 LPS. So I'll just drag this here. Click OK. I can minimize this console in a moment, but if you're ever unsure of what Z-stack you used, if you can't remember, whenever you open up a slice, it'll always be here. So 26Z, 26Z, 26Z. And so that'll be the value that you put in this Z column right here. Just in case you ever forget that and need it, it is in each slice. So we're just going to look at series 1. This is the first section. Do each one at a time. Minimize this console. Then I'm going to go to Image, Stacks, Z Project. We're going to project all of the entire Z stack we took. I'm going to click OK. So now it's basically superimposing all the nuclei imaged in this whole 25 micrometer thick slice. So then I'm going to go to Image, Color, Channels tool. Keep that open for the channels. So we have the nuclei here. The second channel is our microglia. The third channel is our CD68. And the fourth is our blood vessels. And so what we care about right now is first counting the number of microglia. And this should be pretty straightforward because the imaging of the CX3CR1 GFP mice is very clear, so we can really clearly see these cell bodies. And so the way that I count the microglia, I click on this multi-point tool up here and just click. And then I can tap on the screen every time I see a microglia cell body. So that's 1, 2. And the way you verify this is by looking at the nuclei. So I'm going to click Color Composite. I don't need the CD68 or the blood vessel channel, but here, basically to make sure that what I'm counting is a microglia, I want there to be a nuclei completely overlapping with the green. And so I can kind of toggle back and forth and see that exactly where this green is, there's a nuclei. So that is a nuclei. Same here, same here. So we'll use the nuclei marker just to help us with that whole process. But basically, so we count 1, 2, 3, 4, 5. I accidentally double tapped, so that's why the numbers are a little off. 6, 7, 8, 9, 10, 11. So it says 13. It's just because I double tapped. And if you're unsure of how many you counted, you can double tap here, and it'll say 13, which again, double tap, so it's actually 11. So then I will go to my Chrome, and I'll say for mouse 1, section 1, I will type in 11. And see, since I already put in this stuff, then the density factoring in the area is this value right here. And how did I get the area? So that is something that you get. It's based on the objective you use. So we use the 63X. So when you open the slice, you can click on your keyboard Command M, and that's going to take a measurement. So the area of this whole entire square that we're measuring is this right here, 34,000, blah, blah, blah. And that's in micrometers. And so I already have that right here for all of the squares we will calculate. But that's how you do it in the future. So now I can just close this out. Now I'm looking at this same slice. I'm making it a stack. And now I want to look at CD68. So this is the CD68 channel. I'm going to make it magenta so I can see it easier. So you can see here there are these puncta, these dots, but there's a lot of little background noise. So I'm going to go to Process, Subtract Background. I'm going to click OK. And I'm going to click No because I just want it to subtract the background for this specific channel. So that helps a little bit to get rid of some of the backgrounds. Another thing you could do is image adjust brightness contrast, and you can mess with the brightness and contrast to also eliminate some of the backgrounds. But the thing about that is you want to make sure you're consistent for each slice, so you don't want to alter that too much to where it's not the same for each slice, and then you lose the normalization. So I'm going to click Composite here, get rid of the nuclei, get rid of the blood vessel staining, and now we're looking at microglia co-localized with CD68. And so the way that I do this, pretty straightforward, is I can make it a little bigger. I kind of toggle between the two channels, but to help visualize it, but I want to see microglia cell bodies that have pink dots basically overlapping with them. So let's look right here. This one, you can see these pink dots very clearly on the green. I'm going to click off the microglia channel and say, okay, I can see these dots very clearly. And then when this guy is back, I can see clearly that they're superimposed on top of each other. And so I'm going to count this as 1 using that same tool. And you kind of toggle back and forth. Like here, you can see these very clear pink dots, and sometimes the green obscures them, so that's why you kind of want to toggle back and forth. So I'll click on this channel again. That's 2. And I already kind of did a preliminary overview of this and analyzed it myself, but I want to see what you get and if we get the same stuff. But the pattern I noticed is that for the IT LPS mice, almost all the microglia have CD68 overlapping with them. So you should see a lot of pink overlapping with each microglial cell. Whereas for the controls, I only noticed it was about, ranging from 50 to 75% of the total microglia on the screen had CD68 co-localized with it. But you might see something different, but that's just what I noticed. So I'm going to keep doing this toggle back and forth. Okay, there's 1. And these, some of these are pretty clear, but sometimes it's not as clear, and you kind of have to be subjective in your measurement. So like for instance, this microglia here, when you look, the pink dots right here aren't as bright as, say, like these guys right here or these guys right here. It still looks to be like there's CD68 puncta here, so I would still count this, but sometimes you just have to use your judgment because you basically want kind of equal brightness across, and you don't want to accidentally count any areas that where the CD68 isn't as strong. So yeah, then I'll calculate all of that and put in on the second spreadsheet, put in whatever number that is. So let's say it's 8. I didn't finish calculating, but we'll see. And it'll spit that out for you. So that's how I do the CD68 quantification. I'll close this out and I'll show you one of the control brains just for reference. So M1 control, section 1. We will make this a stack. Okay, so first I'll count the microglia. 1, 2, 3, 4, 5, 6, 7, 8. And that seems to be all. So I'll go here. Now we'll go microglia. Let's find M1 control. 8. Now let's go back, and I'm going to close this out to reset the counting and just open up that same exact slice. Unfortunately, that's how you reset the number that you've calculated. So I'll open it back up, and I will make this magenta. Process, subtract background, okay, no. And now I'm going to go here, composite, and I just want these two. Make it a little bigger. So this is the control mouse. So they still have some CD68. So right here, this guy, 1, I'd say that's 1. This is 2. You can definitely see that pink in there. This is 3. This is 4. 5. 6. Now for this guy here, see how these little dots kind of look like background? It's not nearly as bright as, like, say, these guys, all the other puncta we're seeing. So I would not count that. I don't think this one, I think that's just background. I don't think this guy has CD68. Same for this top one right here. Like, I'm not seeing any right here, so I would say that's a no. I think that dot there is autofluorescence. So I think the total that we have here is 6. And so I would put that in 6.